sample size methods for adaptive clinical trials. And the focus here is primarily looking at adaptive trials where sample size is important, primarily in the sense that sample size is going to be changed within the course of the trial, and that's something that you're going to make an adaptive decision of, and the approaches and mechanisms available to do that, and what criteria are important for doing that. And this will be done using the latest version of nQuery, using the adaptive or adapt module that will be available in our autumn release at the end of October. So before we get started, I'd like to introduce myself. My name is Ronan Fitzpatrick. I'm the head of statistics here at StatSolves, who develop nQuery for sample size. I've been the nQuery lead researcher since nQuery Interim 3.0, which I believe will be 5 iterations ago after the autumn release. And I've also given a tutorial series at the FDA for nQuery, and I've also given various other talks, both online, such as this, but also in person. For example, just completed a small intro to sample size course there with our local university, UCC. So in terms of what we'll be covering today, the first section will just be kind of a look at adaptive design, sample size, and in particular, we'll probably be focusing mostly on here today on sample size re-estimation. So the hope here is to give you a background of the kind of pros and cons of adaptive design, why adaptive designs are something which is increasing, and hopefully also give you an insight into the regulatory kind of background that's going around adaptive designs as a potential solution to obviously the overarching problem in clinical trials, in particular of increasing costs and lower success rates. And then I'll look at 2 particular types of adaptive design. One is a blinded sample size re-estimation type of example. There'll be 2 examples there, just kind of showing a very easy approach that can be used for sample size re-estimation where blinding is retained. And then the third section will be about unblinded sample size re-estimation and looking at its relationship, or at least in the way that it's usually done at the moment, based on conditional power and basically as an extension of your traditional group sequential design, which obviously is kind of the oldest and probably most commonly used type of adaptive trial still. And obviously at the end then there'll be some discussion and conclusions, and then a Q&A at the end for any questions that come in during the webinar. Just to remind you, if you have a question, use the questions tab of the webinar software and I'll be happy to do that. If there's any audio or technical issues, you can also use that tab to let me know about that as well. So just in terms of the examples, so you're aware what we'll be doing beforehand, there'll be 2 blinded sample size re-estimation for the 2 means and 2 proportions inequality case or superiority case, depending on what your preferred terminology is. Then there'll be a very brief overview of a 2 means group sequential, which is more just really an opener to how conditional power can be used to do an unblinded sample size re-estimation. So I will mention right now that the unblinded sample size re-estimation method available in nQuery is currently still under construction and development, so I'll be kind of doing a manual show of that, but I'll have screenshots available of how that will work in the new updated version of nQuery at the end of October. Obviously, just for context, for anyone who maybe not be aware, this webinar is being put forward by nQuery, which is a very widely used sample size software in the clinical trials arena. And obviously, just a small statistic here about nQuery, it's very widely used. 90% of organizations with clinical trials approved by the FDA have used nQuery for sample size and power calculations, and some of those companies are outlined here, just so you're aware that that is who is sponsoring this webinar. So in terms of sample size determination and adaptive design, so it's probably useful to kind of give a kind of broad, it's probably useful to give a very broad context here. So obviously nQuery is a sample size determination software. So mostly what we'll be focusing on here is the planning of an adaptive design. But obviously we recognize that adaptive designs necessarily involve monitoring and some information, but the primary emphasis here is on planning and sample size in particular. And obviously, traditionally sample size calculations are based on statistical power, which is related to null hypothesis testing. But obviously, just to mention that obviously you can do it based on other criteria such as precision of a confidence interval width or cost-based criteria, or some mix of the above. And the general idea behind this type of planning is that you're trying to balance those ethical and practical issues that exist from the fact that we're doing a clinical trial. Obviously, a clinical trial is trying to find an innovative treatment or intervention which will hopefully make people's lives better in the long run, and we're trying to balance the kind of Goldilocks problem of having a design which has enough people to be able to make valid statistical inferences, so that it has sufficient power, for example, to be able to say with a good chance that a meaningful, like a significant p-value is actually meaningful in terms of what it's trying to say about the hypothesis of interest, but also the practical issues to come, that you don't want to expose too many people unnecessarily during your clinical trial to what is still an unknown treatment or intervention, because we recognize that in a clinical trial, even for drugs which do end up being successful, they may have side effects which are significant, and obviously exposing people unnecessarily to that risk when they could get the current standard treatment is a major ethical problem, but also has major cost implications, because if you in any statistical scenario, you could in theory just say, well, just take the entire population and give them the intervention. And that occasionally happens in vaccine studies. I've seen examples of that done but obviously that's very expensive and very unwieldy logistically, and that means that trial will never happen and that intervention would never get pulled forward. So you're trying to balance the ability to get a valid inference with the ability to look at the practical constraints that come at running any trial or study. And then there's these kind of, in the worst case scenario, this is something I emphasize a lot in the UCC, the university course, probably less common in clinical trials, which have a very high standard of design, at least usually. You have these type M, type S errors, these magnitude errors, these sign errors that Andrew Gelman talks about, where basically the sign of the effect is different from what you found in your study. So, you thought your study told you had a significant effect, a positive effect, but it actually had a negative effect in the population. That comes in these kind of worst case scenarios where you're under power. But of course, we know that in clinical trials, specifically when we focus on this, the problem is probably more on the upper end of the Goldilocks problem in terms of the cost, basically. And we know that a very small percentage of clinical trial pathways coming from like early stage to phase 1 up to phase 3 and up to approval in phase 4 make it and are successful. Now, some of that's just natural variation. Of course, some of these pathways aren't going to work out because we're trying to explore, scientifically a wide set of potential new treatments, and some of them just aren't going to work because they don't work. But obviously, some of this isn't just that. Some of this is coming down to the fact that many of the ways that we choose to do clinical trial development may increase costs when they don't need to be. And one of the ways that might happen is that in the traditional clinical trial, what happens is that you have to make an upfront commitment to a very large trial to find the effect of interest. Maybe this effect is smaller than, like, you have to power for like the least meaningful, like the smallest meaningful clinical effect, even though you might think, well, this larger effect is probably what we actually expect. And that's one of the aspects that can lead to these kind of failed clinical trials, and particularly those failed clinical trials that either don't get off the drawing board in the first place, or those which fail to be able to find the effects that might have been an interest, but which trying to cost for those kind of smaller clinical effects just wasn't feasible at that point. And of course, adaptive designs is only put forward as one small part of the potential to reduce costs. And there's probably just a certain amount of cost escalation that's due to the fact that we have higher standards of evidence expected for treatments and drug development these days than we would have had, say, 50 years ago. But I think it's fair to say that the methods used in clinical trials have been static for a very long time, and that there's definitely been pressure to try and look at these alternative designs as ways to find these things, especially in the context of things like lower replication rates, in the context of things like personalized medicine and other situations, to control the things that matter to us as statisticians and as trialists in terms of controlling our errors, while reflecting reality that if trials are costing millions and millions and millions, then the incentive to do research and development is less. If at the end of the point you end up having a situation where even if you do have a successful treatment, it doesn't actually get approval. So in terms of what an adaptive trial is, I'm trying to keep it all as low level as possible, so I haven't assuming any pre-existing knowledge. So hopefully anyone who's kind of familiar with the area, hopefully this isn't too slow for you. But adaptive trials are basically any trial where a change or decision is made to or about the trial while it's still ongoing. And so obviously that encompasses a wide variety of potential adaptations. And this is just a really small selection on this slide. But obviously the earliest type of adaptive trial, a group sequential trial, had early stopping, which is where you can stop the trial given that the evidence is really strong, either against or for your proposed treatment being effective based on the evidence so far. Sample size re-estimation, which will be the one we'll be focusing on today. But obviously, we're looking at a huge increase in the variety related to enrichment trials, where we choose to kind of keep all the groups, but perhaps increase the sample size in some specific groups because they're the ones which are most promising. Seamless designs, where we can include information from earlier stage trials, say stage 2, and then use that information seamlessly within the context of stage 3. And then stuff like adaptive dose finding, because obviously we know that dose finding is a very common objective in early stage trials, and having an adaptive approach to that rather than having to make all these assumptions up front. And I think adaptive trials are basically very quickly changing the way that early stage trials are happening. But I think that's also filtering up to these confirmatory trials, phase 3 trials, as well as, that's where the majority of the cost is. So I think, the proliferation of these adaptive trials is also coming with the proliferation of stuff related to Bayesian statistics and Bayesian inference as well, especially at early stage, but it's having an effect on larger trials as well. And that makes sense because so much money is at stake. 90% of the cost is at the phase 3 side, so it makes sense to focus on that as well. And the hope here is that by giving more control to the trialist over the trial, they can improve it or adapt it to ensure that, based on the available information, they can maximize the chance that this trial can do what it sets out to do, which is make the best and most accurate inference about what matters. So, in early stage trials, that's going from, we have 20 potential treatments. We really want to winnow this down to like the 4 or 5 that might be relevant for later stage or investigation. That's an adaptive trial, makes perfect sense there. But maybe you just want to keep that information rather than just throwing it away and starting a new trial. And then later stage trials, you might have a situation where you initially powered for an optimistic design. So you ended up being more optimistic, but there was still a potential clinically relevant effect that you haven't powered for, but that you could save from that situation by using enrichment or using sample size re-estimation. And the hope here is that, on a macro level, adaptive trials can just decrease costs by reducing the number of failed trials, but also by, also making sure that trials can adapt. Trials don't have to be a one-size-fits-all Procrustean in bed, where you kind of, if you're really interested in a drug, you don't have to just cover every single possible scenario. You can adapt to the scenarios as they show up, the fringe cases as they come up, rather than having to kind of pre-plan and put upfront investment into those scenarios, which could happen, but maybe aren't as likely as the ones that are of maximum interest to you. So in terms of the pros and cons that come with adaptive trials, the advantages should be fairly obvious. One is that, particularly in group sequential designs, you have the ability to make earlier decisions. So you can stop a trial early if there's really strong evidence. And that's obviously not just useful because obviously, it'll reduce costs, and it'll also decrease the amount of time that it takes for these drugs to go from trial to store shelf. And that's important, but that's obviously not just important to trialists and to companies who develop drugs and other interventions. That's really important to patients. Patients are increasingly being placed at the center of of clinical trials, of why they're justified, of how they're conducted. And I think patients are one of the strongest advocates for adaptive trials because they want to see decisions happen earlier. They want to be able to make decisions within the trial, but also in terms of getting that treatment to them, on an official basis or an approved basis as quickly as possible. And so I think, the adaptive trials have a really strong selling point in terms of being able to make earlier decisions, either directly in a group sequential design, or just because the trial can be increased in sample size quicker if you need to, if say, recruitment rates were slower than expected. Obviously, I talked about the reduced potential cost and the higher potential success. That just comes with the fact that, you're kind of hopefully, if you knew what was going to happen beforehand, you might have an idea of what the trial would have looked like in that case. The idea behind an adaptive trial is hopefully that you'll have the decision-making process available to move from, your initial trial, which had maybe some mistakes or mistaken assumptions, to that optimal trial if you had known a priori exactly what was going to happen, and that process will, move you from, almost to the platonic ideal, say, of what that trial could have been. And hopefully adaptive trial will give you the best opportunity possible to get closer to that platonic ideal. Obviously, greater control is a big thing, and these seamless designs are a very strong emphasis now, because you could very much argue that traditional clinical trial development can be quite wasteful in the sense that each trial is treated completely separately, but all that information which has been used previously is being thrown away, particularly within a frequentist analysis. In a Bayesian context, maybe it's not quite as true, but even if you're using Bayesian, you're still using that only to develop your prior rather than using that information directly, depending on if you're using well, you might be using empirical prior, which closes the gap a bit, but nevertheless, you're throwing some of that information away in some level rather than using it directly. You're losing some of the degrees of freedom that come with using data in its rawest form. But in terms of the disadvantages, they're more complex. Obviously, that means there's a higher requirement in terms of technical expertise, statistical expertise, and such. Obviously, for many of the people who are involved with larger drug development companies, that's not an issue. But obviously, for academics or for CROs on a tight budget, that may not be as true. So obviously, there is additional work. But obviously, as certain types of obviously, there's a huge variety of adaptive trials out there because it's an area of really active research interest. But obviously, we expect that as time goes on, this will series of adaptive trials will be winnowed down to the ones that work and the ones that are practical, and then software solutions like nQuery, but like other software, will become available to make that process of doing that adaptive trial easier. So in phase one, CRM, continual reassessment methods, which can be considered a type of adaptive trial from a certain perspective. 10 years ago, the methods were developed in the early 2000s, but 10 years ago you wouldn't have a software solution. Now there's plenty of software solutions out there for planning and analysis, and obviously that's just natural development as time goes on. For clinical trials specifically, especially confirmatory trials, the logistical issues are probably one of the biggest things to keep in mind. And basically, I have IDMC here, the Independent Data Monitoring Committee. If you're doing an adaptive trial in a phase 3, which is unblinded, which is to say that the adaptations are being made based on the unblinded data, which we'll see in the sample size re-estimation example later on, the Independent Data Monitoring Committee becomes way more important because they're the ones who will end up making the decisions based on the evidence. And this is true of group-sequential designs, even the most basic type, well, one of the more basic types of clinical trials. But as these adaptations become greater and there's more variety, the IDMC becomes even more and more important as time goes on. So in terms of, logistically, that's something to really keep in mind. Now, I won't focus on that too much here because it's a complete different thing itself, but it's something to keep in mind. Obviously, just to mention that more complexity also means greater expertise, and you may also end up using modified test statistics which aren't directly relatable to kind of the traditional test statistics you might have used in the fixed term trial. So that can, some people mightn't like that, for example, particularly if they're using weighted statistics, which end up kind of engaging in non-intuitive ways. And you'll see an example there. I'll mention that during the example of the unblinded sample size re-estimation of one of the alternatives where people aren't weighted equally. They're weighted differently depending which cohort they're in and what that might mean. And then there's just, excuse me, then there's just this final point, and this is probably the one which has the most debate and interest, and that's regulatory approval. And I think it's so important. It's probably just having a kind of summary by itself for that. So in terms of what the regulatory background is right now for adaptive trials in clinical trials, most of you may be aware that the FDA, their Center for Drug Development and Biologic Development, issued draft guidance in 2010, and the main thing that seems to come out of it in terms of, I wouldn't say controversy, but in terms of why people might have felt slightly less confident of doing adaptive trials based on it, was this distinction between what they called well understood trials and less well understood designs. And so there's been a lot of talk here recently about, since the 8 years since then, about whether that's been a useful distinction or not. And the EMA had a similar reflection paper in 2007, which was a bit more liberal, but the reflection papers tend to be not quite as in-depth in this particular case. But what's happened since then, since 2010, is that there's been a massive increase in the interest in encouraging adaptive design. And as I said, some of that's coming from trial sponsors themselves, but a lot of that is also coming from patients themselves being really interested in this area of adaptive design and really wanting to encourage these designs, which give like more power to getting these drugs approved earlier, but also even the participants within the trial have a bit more influence over how it goes, how the actual trial is working, while obviously retaining, the overall objective of having a statistically legitimate inference at the end of the study. So in the US, we have the Innovative Cures Act, which was passed last year as a budget, like as part of the budget bill. And in the EU, we have this Adaptive Pathways program, which was ran as a pilot, I believe, from 2013 for 3 years, or maybe 2014. But the big thing, and like I would say that we're looking at adaptive designs here. There's a new draft guidance on adaptive design expected from CBER and CEDAR later this year. I think the end of October is kind of when people have been talking about, and that is expected to be much more in line with what we're seeing with this Innovative Cures Act in particular, in terms of encouraging adaptive drug design. So we might be on the threshold of a huge proliferation of adaptive design for those who are interested in doing it. So, for example, just a couple of weeks ago, the FDA issued guidance on an expansion cohort design for a rare disease. So that's just an example of that's basically a type of seamless adaptive trial, and I think that's just kind of a kind of flavor of what's to come. So for anyone who's interested in the regulatory background, hopefully now you can have the Cliff Notes version of it here. Obviously, it's just kind of based on what I've seen in talks and stuff like that. But obviously, if you probably want to talk to the relevant people at the FDA and EMA to get this. But this is my current opinion or understanding of where things kind of stand on the regulatory side. So as I said, obviously adaptive design, each type of adaptive design, seamless, enrichment, etc., could basically be an entire webinar by itself. So for this one, we're just going to focus in on sample size re-estimation. Firstly, because it's a nice introductory one, because it's pretty easy to understand. So hopefully anyone who hasn't used adaptive design before won't get too confused at this point. And it's also one which it kind of makes sense to look at, because this is an adaptive trial where we're focused on increasing the sample size if it was needed. So basically, this kind of is an obvious adaptation target, because when we're doing a sample size determination, and if you come to any other webinar or talk to anyone about sample size determination or read any intro book, intrinsically there's uncertainty in a sample size determination, right? So we're making assumptions about what we think is going to happen beforehand, and then we're like, if that's true, here's the sample size required to achieve a significant, like a power of 80%. If we're doing this beforehand, if this is at the planning stage, then it makes sense that some of those assumptions are going to be wrong or different, and thus it makes complete sense that if we have information midway through the trial or as the trial is ongoing, we could use that to basically update what our prior belief should have been and ensure that the trial kind of moves closer to what it should have been in the first place, to reflect the true value for the variance, the true value for the effect size, or the true value for what other parameters of interest. Just to mention here, obviously, this is kind of more suited to where there's like short follow-up or where we know when things are going to happen, because obviously if you have like a 12-month follow-up and stuff like that, then information kind of becomes outdated quite quickly. So it's just a small note there. And just to also mention that this could also be used to lower sample size versus your initial sample size, but I would say within the regulatory context, decreasing sample size versus your initial sample size estimate, I wouldn't say it's a no-no. I would just say it's something that you'll have to justify to a greater degree than the idea of increasing your sample size adaptively to new information. And so, as I mentioned here, there's two primary types. There's blinded sample size re-estimation and unblinded sample size re-estimation. Blinded sample size re-estimation is one where you don't need to unblind the data, so you don't need to find out which group an individual is in to improve your sample size estimation, whereas unblinded does require you to do that, basically because they're targeting different aspects of the assumptions required for a sample size determination. And the basic version is that the blinded sample size re-estimation is usually targeting a nuisance parameter, such as the variance or the control proportion, whereas the unblinded sample size re-estimation is actually targeting the actual effect size, so the effect that's actually happening at the interim analysis. So we're going to cover each of those in turn. So in terms of blinded sample size re-estimation, what we're looking for here is to use an interim estimate of the nuisance parameter which we can basically, like, we won't be able to get the direct value of this usually, but if we're willing to make a few basic assumptions, we can basically get a reasonable, a better estimate of this than we had before we started the trial. And obviously the idea here is, don't let the perfect be the enemy of the good. If we get an estimate that's 50, 70, 80% better than the estimate we would have had at the start when we had we had less information to work on, then that's a better way to do your sample size and improve your study as it's ongoing, rather than just, oh, well, we made a mistake at the beginning, we have to just live with that, which is kind of the classic situation that occurred. And just to note, this is what would have been right now already considered a well understood type of adaptive design. So if you do a blinded sample size re-estimation, you usually won't have too much issue on the regulatory front, as long as it's done well and you have the sufficient safeguards to ensure that the blinding is retained. And obviously, like, blinding can be retained is kind of a default part of clinical trial, and people are exposed to the real data on a blinded basis all the time. So the barrier shouldn't be too great here. There are multiple methods available, but I'd say the one that's probably proliferated the most is what's known as an internal pilot approach, primarily because it doesn't really require changing that much. It's literally just taking, say, the first 20% of your data, treating that as if it was a pilot study to find this nuisance parameter, say the variance, updating your sample size estimate with this new estimate of, say, the variance, and then continuing on as before, and then using the entire set of data as if the internal pilot had not existed in the first place. There are other approaches, because they're like, I think it's important to note that it's often stated that the internal pilot approach does retain the overall error, so that there's no error rate inflation, but it's probably better to say that the inflation is negligible for the cases which are most relevant, particularly in the inequality superiority testing case. But you're not guaranteed to retain to avoid error inflation. There is a tiny error inflation, but it's negligible enough that you can kind of get away with it. There are other approaches which guarantee you error control, but they kind of fall into that problem of being more complex, so they require a bit more expertise. You can't just get someone who isn't an expert or isn't a statistician, asking them to do a weighted statistic. It's not that difficult, but it's something which might be a barrier that makes them less likely to acquiesce. But it's also a situation where that weighting problem will come in, because if you're using a weighted statistic, that means that the earlier cohort in the internal pilot in the equivalent of the internal pilot, those people end up getting weighted more if you do end up increasing the sample size. So I'm focusing on the internal pilot approach because it's just probably more common, and it's also just easier and just more likely to get used in practical purpose. Just to mention the options that will be available in the autumn release of nQuery, there'll be blinded sample size re-estimation for the two means case and the two proportions case. And just to give the overview here, that the sample size criteria for the means case is the variance, or I should more say the per group variance. And there are 3 different methods that I'll show for what a reasonable estimate for the variance might be. And then for the proportions case, there's the overall success rate is used as the kind of nuisance parameter that you estimate from your blinded internal pilot. But just to mention that, like, I'll go through a certain example, but just to mention obviously that to get the individual proportions, you're making some assumptions about, you have to continue to assume that your effect size is true. So this is just a very simple t-test example from the literature, from the New England Journal of Medicine, from a paper from 2006. So what I'm going to do now is just very briefly show how a blinded sample size re-estimation will work in nQuery within the new adaptive tables. And you could basically do it right now if you went to the classic 2-sample t-test table. You entered your difference of -9. You 16, and then you entered your power of 90%, and you got 68. So just like, this is replicating the original example, and then you can go, well, the new estimate is like 18 or something like that. But the problem with that is that you basically have to come back to an old problem like this, and it also means that it's not very clear if you're trying to communicate this to someone what this would actually look like, or trying to save something for later use in terms of doing a sample size re-estimation. It also doesn't elucidate the variance estimates that could be used in this scenario. So the first thing we're just going to do is very briefly replicate that example. So I've just assumed 0 and -9 to just get back the difference of means of -9 here, just like a small shortcut. And you can see like these are all the same rows as the two sample t-test. So the first thing that we're doing here, for obviously for a sample size re-estimation, is that to re-estimate something, first you obviously have to estimate it. So what this is really just doing, these first rows, is replicating basically MTT0 here in this table and getting the initial sample size. Note that in the context of sample size re-estimation, we usually talk about the total sample size rather than the sample size per group. That's just a small note. Just for anyone who's not familiar with nQuery, that should be like the minority here, just to know that each one of these rows is an assumption required for the calculation. Each column is an individual calculation, and as we select rows, you'll get a little help card on the right explaining what the that row means and what's happening here. So what's happening here is that we're like this is doing the same thing. We have all these are the same assumptions we had to have previously. We're not going to be able to update our difference in means assumption because this is going to be blinded data. That means we're not going to see who's a group 1 or group 2, so we're not going to be able to see what the new effect size might be. But we are going to target this common standard deviation. We had an initial estimate of this for 16 in this case, and now we're like, well, what if this was wrong? What if the variance was higher than we expected? And so what I've just done here, like I've done a little simulation where the assumed variance in each group was around 20, and then that outputted 20 values here. So we've assumed that the first 20 people, so 10 in each group, are going to be in our internal pilot study. And what's going to happen then is we're going to just take those values to create an updated value for the initial common standard deviation. So once we click on the internal pilot sample size, we'll set this to 20, and that's equal to 10 per group, basically. And what will happen in nQuery then is that you'll automatically get this blinded sample size re-estimation side table, and this is a pretty simple to understand. What we have on the left-hand side here is 3 options for the variance value that you want to use. So Friday and Keyser, who are probably the biggest advocates of the blinded internal pilot approach, they actually say that if you just take the 20 values that are from each group, you can more or less ignore the between group effect. So there's if you look at the total variance, you can split it up into the within group effect, within group variance. So that's the variance due to the amount of variability within each group, and the between group effect, which is the amount of variance for the total variance that's affected by the fact that the mean in group 1 is 0 and the mean in the other group is -9. And what they would generally say is that amount of bias for a reasonable sample, like for any sample size that's greater than like 2 or 3 or 4, like once you get up to like 10 or 20 even, you can pretty much ignore that, and you can just use the variance estimate itself, or I should say the standard deviation estimate directly with no adjustment, and that's fine. And they say that actually works really well, and that's what the simulations generally seem to show. But if you want to look at other alternatives, there's the bias adjusted variance by Zucker, which just basically tries to give you, like, basically you can actually derive fairly easily the expected bias for the within group common like standard deviations or common within group variances given a fixed effect size. And what he just does in Zucker's case is that, okay, assume the effect size is actually the effect size that you set up here. So the difference is actually equal to 9, and then calculate what the variance would be, the total variance minus that bias term due to that effect size, if that effect size is the true effect size. And then the final one is just the kind of a very similar to what you might do if you had an external pilot study. You might be encouraged to use the upper confidence limit for, say, the 90% or 80% upper confidence limit as the actual value that you then use for your sample size calculation. That's quite a common like rule of thumb used by Julius and other authors, and that's just basically using the same approach but for an internal pilot estimate instead. So the 95% here is just because it's kind of very familiar, but I would say actually an 80% confidence level is probably the one. I think that's the one that's usually encouraged for, because it's 95% just gets you very high, and you could have much higher sample sizes than required. Obviously, all of this comes with a trade-off that if you it comes with a trade-off that you'll have a less likely of underpowering your study if you use the upper confidence limit, but you obviously have a higher chance of overpowering it to a certain extent, with overpowering in the sense that higher sample size than necessarily would have been required for the given truth that existed, -9 difference in this case. And you can see here that the three different values are provided for reference. directly by the software, and then the one that's actually used in the main table for the re-estimated sample size is the one that's just displayed here and then moved into the new into the main table. And so you can see that reflected here. If I select this option here, this is automatically updated. And what happens then is that you then get you basically get a re-estimated like idea of what the total sample size should have been if this estimate is true, and this estimate was, this is the true estimate now, around 18 was the common standard deviation estimate in this case rather than 16. You have an increase from 136 to 168, right? And then it also just provides you that given that the internal pilot exists, so there's 20 people that you already have data for, these are the two values that exist for what you might want the rest of the study to be. So, that basically just equal to this minus this. And there's two approaches you can do. You can have the restricted version, which means that the minimum sample size you're willing to allow is equal to this initial value, n here, of 136. So even if the variance, even if the variance decreased, for example, you still want the initial total sample size to be your sample size in the study. That's what the restricted would be, whereas the unrestricted, the minimum value would be equal to the pilot study. So imagine that this variance estimate was, sorry, the standard deviation estimate was way too high. Imagine it was like closer to 1, for example. Obviously, that'd be a major problem for planning. But if that happened, maybe you only would have required 20 really in the first place to get that estimate, and in that case just set it to, the pilot study is good enough for this effect, for this variance, so we could just stop here. I would say the restricted one for regulatory purposes will often be preferred, just because of that there's kind of a bias, I would suppose, against using like reducing the sample size. And of course you could explore this further by say, you could take that internal pilot standard deviation and you could say, well, what would the effect on the re-estimated total sample size be if that had gone from, say, 16 up to 20 in increments of 1. And so you could see here, you could have this little graph here showing that, there's a very basic relationship between the internal pilot standard deviation increasing and, the expected total sample size. So this is a total sample size here, and obviously you just need to take away the internal pilot size to get the, the estimated rest of study sample size. So like the main thing to take away from that there is that, if you want to do a sample size re-estimation, you can do a blinded sample size re-estimation for nuisance parameters like that, especially in the means case, using a very simple approximation. Like, you take 20 people, you take the first 20 people, you find out the variance or the standard deviation of those people, and then you update your sample size calculation with that. Very simple idea, and you can do that without having to do adjustments or weighted statistics or anything like that. And you can be, with a high degree of confidence, especially in an inequality case, believe that you're not going to have a very large error inflation. Just to note, things are a bit more complicated in the non-inferiority and equivalence case. Error inflation tends to be a bit higher there. Don't really have enough time to go into it here, but conceptually, it kind of comes down from the fact that the null region of non-inferiority equivalence hypothesis is kind of centered around 0. Like, you're often assuming that the effective interest in non-inferiority equivalence is actually equal to 0, and that would mean that your blinded estimate would actually be very, very close to the true estimate of the between group standard deviation, because you wouldn't have that that between group variance effect wouldn't exist if they're both they both have the same mean and same standard deviation. So it's conceptually actually leads to higher inflation in those cases. So what I want to do now then is just another example for the two proportions case, and the basic idea is hopefully here to show that it's very, very, very similar. So this is just a study from taking up endoscopy for a type of surgery using pancreatic papillotomy, and once again, there's here's the actual segment used for the sample size calculation here, and you can see here that actually they assumed in their sample size calculation it'll be 60 and 90% respectively, but that the actual effects from what they were basing it on previously was closer to 89 and 64. So that's it. But like what we're targeting here is just we're gonna take the first like say 20 people, and that's a larger cohort obviously in this case, because previously we were taking the first 20 people out of 136. Now we'll be taking the first 20 out of 64 or so. And obviously, it's worth noting, of course, that the larger the proportion that you base your nuisance parameter estimate on, the more accurate it will be, and the more the lower the over or underestimate versus the reality will be likely. So in terms of what this looks like, once again, it should be very familiar. These first set of rows look very, very similar to the number of rows here. And basically, we could quickly replicate the example used in that example. So we have 0.05 at the two-sided level, 60% in group 1, 90% in group 2, so a 30% increase, odds ratio equal to 6, very large odds ratio. So obviously they're very confident in this potential intervention in surgery, and it's 32 per group, or roughly 64 in total. And what you'll note here is that the first set of inputs, we can literally just kind of copy and paste them across with very little. We'll just copy and paste these across. And you can see here we have the difference in proportions and odds ratio just because the method put forward by Frieda and Kaiser in this case uses the difference in proportions, but the odds ratio is useful information and is inherited from the PTT0 table that we kind of use as our standard. So the solvers being used are inherited from this table. So in this case, they wanted 80% power and a sample size ratio of 1, and they had a total sample size of 63. So this is slightly smaller, and that just reflects the fact that when the sample size ratio is equal to 1, you can always just assume it's equal to 64 and force it to be the same. But in many algorithms, what they'll do is that they'll go, well, increase each in increments of 1 and pick the one which actually is the lowest possible to get you above that. So the ratio is slightly off 1 here, but obviously, for practical purposes, you might increase this to 64, but we'll just have it at 63 for now. So once again, we'll set the internal pilot sample size to 20. And in this case, we don't really need a side table because there's only one piece of information that we'd really need to get an estimate of the total success rate, and that's the number of successes that we had. And in this case, we were assuming that the success rates are 0.6 and 0.9. So if we were to have an estimate based, if that was true, obviously the expected success rate in that case would just be halfway between them, given that we have equal sample size. We just take 0.6, we add 0.9, we divide that by 2, and we get 0.75. But let's assume that the pilot proportion, the overall number of successes was closer to, say, 0.7 instead of 0.75. And that might be due to either the control group or the treatment group proportion being lower than we expected, but we know it's slightly off. But assuming that this difference was kept constant and these were basically both shifted down by 0.025, we can estimate that the sample size would have increased from 63 to 71. Not too big a difference, but obviously for a study which only had 63 people in the first place, you may consider that to be substantial, because obviously surgery isn't a trivial thing to recruit people for. And in that case, the restricted and unrestricted sample size become 51 each. So you can see here that, if you have a nuisance parameter that you can estimate beforehand without unblinding, this process is pretty simple. So the last 15 minutes here, I'm just going to spend on the unblinded sample size re-estimation, which is probably what right now is kind of being somewhat widely used, but probably would still be considered within the remit of less well understood. But obviously that's subject to change, and it probably is very likely to change in the very near future. But right now, unblinding is just one of those things that people don't like. But I would say it's the reason why unblinded sample size re-estimation could probably be called in the well understood, with a bit of justification. It's really just an extension of the group sequential design. So the group sequential design is one where you're doing an interim analysis at pre-specified time, say halfway through, and you're making one of two decisions, or possibly one of at least two decisions, to either stop the trial early, say halfway through, for benefit or futility when the evidence is really strong for or against stopping because of benefit or futility. And the way that we adjust for the multiple analysis in the group sequential design is by basically doling out our total error, like say our alpha error, our significance level of 0.05, like we put a certain proportion of that at look 1 and a certain proportion at look 2, and then make the various adjustments required to ensure that we retain our overall error rate as the same as for the fixed trial, and obviously increase sample size, etc., to reflect that. And then we can also spend, so that's for the efficacy bound. So if we want to stop early for efficacy because we have strong evidence for our study intervention being good. And then there's the beta error where we want to stop for futility. And I would say that stopping for futility is very probably the most commonly used adaptive design right now, having a basic futility decision halfway or somewhat through your trial. Very, very common, very, very widely used. Just to mention that in the autumn release, we'll also be updating our Group-Sequential Design suite to provide for futility-only designs, some additional outputs related to parameterizations and the sample size in the looks table. And obviously, we'll be looking at new tables for Group-Sequential Designs for 2-sample survival, accounting for accrual, and 1-sample means and proportions. And obviously, we'll be increasing the options in Group-Sequential Designs as time goes on as well. And just to mention here, we have the error spending approach. This is just a theoretical slide for that. I'm not going to go over this, but if you're interested, we have various videos on this topic that I've done previously. So just what I'm going to briefly do here is replicate a group sequential example. And I've done this several times before. So if I go through this a bit quick, don't worry, you could get a video of this being done properly previously. But what I'm just going to do is very quickly replicate this. And similar to that idea that I was talking about with the blinded sample size re-estimation, group sequential designs work very similar in the sense that the first set of inputs are basically the same inputs that you would be using in a 2-sample t-test or 2-sample z-test, and similar chi-squared test situation for 2 proportions and log rank test for survival. And so we have our changes from baseline here of minus 2.3 and minus 1.5 for Botox, which is less successful that's expected to be less successful by these authors, and their proposed anticholinergic treatment or therapy for incontinence is the case here. So this is equal, so in this case, we'll just take the default set of interim, like group sequential design. We're going to be using O'Brien-Fleming bounds. This is probably the most commonly used type of bound, and it's a conservative bound, which is to say that it's a bound which spends far less of the error at the earlier looks and keeps as much as the error for the final look. And the reason that usually happens is because the most controversial thing that could happen in a group sequential design probably is that you get an effect size at the end of the study, which would have been significant given that you'd done a fixed term trial, but which isn't because of the error spending approach that you did, because you spent too much error at earlier looks. So even though less conservative methods such as the Pocock bounds are actually probably more efficient over a wider set of situations, the O'Brien-Fleming just kind of reduces the probability of that controversial moment happening where you end up being non-significant for an effect size that would have been significant if you'd just gone with traditional trial. That's my opinion on that. That's just, there's no, you probably wouldn't find anyone officially talking about that in the literature, but that's my read on why people tend to use O'Brien-Fleming. And you can see here you require 109 per group. See that you get a table here, and basically you calculate these z statistics. So if the z statistic is between at look 1, this is halfway through, is less than 3, it's less than 3 or greater than minus 3, you continue on to the next look. And if it's greater than 3 or less than minus 3, like it's outside this boundary between minus 3 and 3, then you would reject you would reject at that point, and you can finish the trial early because you have very strong evidence for efficacy that it would be efficacious if you continue the trial to the end, basically. But obviously, as I mentioned, it's probably more common that you would do a one-sided trial with the same parameters, but instead of using like a 2-sided efficacy bound, because if we're talking about efficacy and futility, a 2-sided hypothesis, I wouldn't say it's not meaningful, but it's kind of weird to talk about. I think practically speaking, especially in a clinical trial where we probably know what success means versus not success, there's definitely one direction for the effect size that would be efficacious, and one which would be futile in terms of the drug getting approved. Just to mention here, you have a choice between non-binding and binding. Non-binding just means that if you cross the futility bound, you can continue the trial if you wish, and I would say it's by far the more common decision than binding, where you can't continue without inflating your beta error or your alpha error. And the reason I would say that is because these group sequential designs are giving you the opportunity to stop early, but obviously there are other considerations from the independent data monitoring committee or from other people for why you might continue a trial. A very common one would be safety considerations. So I would say that non-binding is probably the recommended choice. And you can see here that this would increase the sample size per group by 6 per group. And instead of having a lower bound, we now have a futility bound. And basically, if we had a sample size, if we had a z-statistic greater than 3 at our halfway through, at our interim look, we would stop for efficacy. But if we had one less than 0.56, then we would stop for futility. And stopping for futility is probably less controversial because obviously at that point you're not the trial is, continuing to invest resources in a futile trial is generally not recommended. And obviously at the end point, you either find for futility or non-futility, because obviously at the end point you make a decision about whether your result is significant or not. Just to mention here that the nominal alpha here is the value for the significance level that you end up using for your traditional z-test or t-test without any adjustment. So that's probably the one that you're most interested in, and that will end up being used for what I'm going to do is kind of a manual example of an unblinded sample size re-estimation. So I will note, as I noted at the start there, we like the current unblinded sample size re-estimation and indeed interim monitoring, which we consider to be kind of very related, will be available in the release in October, but we haven't quite got it complete yet. I'll show you a screenshot of what that looks like, but I can actually very briefly illustrate it for you now. But just in terms of the theory of unblinded sample size re-estimation, unblinded sample size re-estimation at the moment is kind of generally associated with the idea of conditional power. And conditional power is basically just the power that you would, like the probability that you would expect to find a significant p-value at the final look, given what's happened so far. And conditional power can be used as a very simple criteria for futility testing. So if you have low conditional power, you could say, well, if conditional power is less than, say, 30%, we'll just stop the trial for futility. It's more flexible than the kind of error spending approach that I've outlined in the previous example, but obviously your error rate guarantee isn't quite as high in that case. I should note that the calculation still requires you to make an assumption about what you think the true difference is, and obviously it makes a difference whether you choose to go with the original effect size you set or whether you choose to go with the interim estimate of the effect size as being what you think is actually happening underneath the hood if you apply it to the population. But what we're using conditional power here is not as a measure of whether we're futile or not, but more a measure of what is promising. So Chen, Deletz, and Lan, who wrote one of the main papers on this type of design, talk about increasing sample size when a result is promising. And promising basically just means that based on what's happened so far, you have a conditional power, which remember is you basically your best estimate of what you think the power would be. The power is given what's happened so far. Is less than your target, so it's less than say 80% or 90%, but it's higher than a certain threshold. So let's say it's between 50% and 80% in this example. That might be the zone, like that might be the area of conditional powers that you would consider promising, and that's actually the area recommended by Chen, Demetz, and Lan. Note there's a very related term of predictive power or Bayesian predictive power, which is essentially just conditional power averaged over a prior. And that's very similar to the idea behind of assurance and normal power. I don't really have time to go into here, but if you're interested, feel free to ask a question. I'll go over that in more detail. So in terms of how conditional power is used on blinded sample size re-estimation, it's basically being used as the criteria for adaptation, which is to say that the value you get for the conditional power at the interim analysis is what you're going to be using to decide what is promising. And if you have a promising result, which is to say one that isn't telling you that you're which is not so high that you could just use your current n and you'd probably have sufficient power regardless, but which is high enough that you don't expect that you have that this trial is unlikely to succeed, that in that case you can increase the n and basically save a trial which perhaps wouldn't get there. And promising is really defined by the user, but it does have certain effects on what statistics can be used. And the general way that this is justified, or where this might come in or be useful, is where you could design a study for, say, a more optimistic effect size up front, and then at a later point, if a clinically relevant effect size was found at the interim analysis, but it's kind of smaller than the optimistic one, optimistic in inverted commas, than you had initially, then you can increase the sample size at that point if you feel, based on the new evidence that you have, that okay, like this is not like it's not quite as good as we thought. It's still very relevant, and it's worth the additional investment of extra sample size to keep this trial going. And there's two main methods put forward, one by Chen, Demetz, and Lan, and one by Cui, Hung, and Wang. And the first is the one I'm going to focus on, just because you can basically use the exact same statistics and analysis as a group sequential design using Chen, Demetz, and Lan if you're willing to follow two constraints. One is that you only do a sample size re-estimation at the penultimate look, and the other is that the conditional power has to be above a certain threshold value. Chen, Demetz, and Lan set that at 50%, but there is additional papers by Mehta and Pocock where you can derive an exact lower bound, like above which, if you increase sample size, you would not expect a large increase in your alpha error. But the Cui, Hung, and Wang approach is basically just a weighted statistic where you weight the cohorts based on the original sample size, and you could do that at any look and for any sample size increase and any conditional power. But the big issue with that is that obviously if I increase the size of the second cohort, for example, but the weight is based on the initial sample size, those people in the second cohort are being weighted lesser or differently from the people in the initial cohort, and that's kind of seen as controversial in certain quarters. Just to mention that the first release of nQuery will be related to two means of proportion. Survival is obviously something we're looking at in future releases. So in terms of very kind of showing how this might work, we can very quickly just do a very toy example here. This is just a screenshot here. We could just do a toy example here. For example, let's imagine that the difference that we found was equal to around, instead of having an effect of minus 2.3 and minus 1.5, so minus 0.15 is our initial estimate is the initial estimate that we expected for the anticholinergic group. Let's assume that our control group estimate was correct at minus 2.3. Those standard deviation estimates were correct based on interim analysis data of 2.1, but that the actual effect for our new treatment was actually slightly less than minus 1.5 change from baseline, and it was actually closer to minus 1.65. Obviously, values closer to zero in this case were considered better. And then what that would translate to is into a z statistic. We can easily calculate that. That's just equal to the difference divided by the standard error. To save time, I'm just going to say that was equal to 1.667 approximately. And then, we know what our target sample size was in each group. It was 115 per group. So we're just inheriting this from our group sequential design here previously in column 2. The significance level is equal to the nominal significance level at the final look, which is equal to around 0.245. So you can take that from this table here. So we'll let this to 0.0245. And then the sample size that we were using, we're going to use half this, like this group design that had a halfway true. So half of 115 is around 58 per group. And in this case, the conditional power based on this interim estimate for the test statistic, the z statistic of 0.1667, where the difference is equal to this, gives a conditional power of 70%. If we assume the actual true effect is, if we assume the actual true treatment effect is equal to this. But we would note that if we were to change that assumption to where the effect was the original effect, so let's assume this is a slight underestimate, but we still think the effect will be the original estimate of minus 1.5. The conditional power would increase in this case, and maybe you wouldn't have to increase. But we could then have a simple rule say that if we're if we're between 50% and 80% conditional power, we would double the sample size. And the reason we might do that versus getting the exact sample size to go to conditional power of 80% is just because it might be possible to reverse engineer I'll do this very briefly just to show that we had a simple rule of increasing, doubling the sample size, we get to 94% power. But if we were to go to the exact sample size required, which in this case is around equal to 145 per group, The reason we mightn't do this of increasing exactly 25% to this is simply because we understand that it might be possible to reverse engineer this. And reverse engineers and get the actual estimate that they used here for the interim effect. And obviously that would undo the unblinding for the actual study participants and trialists, and that is a major no-no from a regulatory perspective. And so there's just a small screenshot here of what it will look like in the final design. If you're interested in the unblinded sample size re-estimation, obviously I haven't had time to go into it in detail. We will be running a webinar specifically on unblinded sample size re-estimation, probably at the end of October or early November. So if you're interested by the concept but you don't have time, you want to see that in more detail, we will be doing a webinar on that topic in the future as well. So just in terms of discussion conclusions, and I just want to apologize, I think I've run a little bit over time here. We're expecting adaptive trials to become more common due to their reduction of costs and the kind of regulatory, patient interest and governmental interest in increasing the efficiency of clinical trials and using these adaptive trials. Sample size re-estimation is a very common type of adaptive trial, and unblinded sample size re-estimation you can almost consider to be a standard part, a standard tool already, and unblinded is growing in interest very rapidly. Blinded sample size re-estimation targets nuisance parameters, usually the variance, and anytime that's underestimated, for example, with the variance. And the internal pilot approach is a very easy way to do a sample size re-estimation, which doesn't involve very many overheads logistically or statistically or expertise-wise. You can basically just do it and use the same statistics as you would have otherwise. And be fairly confident that you now have a better study than you would have previously. Whereas unblinded sample size re-estimation targets the unblinded effect size and can be considered an extension of the group sequential design. And thus, the additional logistical issues to come with logistical designs are kind of included within an SSR, unblinded SSR design. And conditional power is being used in unblinded sample size re-estimation as our ability to define those studies worth investing that extra money involved in recruiting the additional sample size. Just for anyone who's interested, this was the 2018 release. 72 new tables were added, 52 core tables for epidemiology, non-inferiority equivalence, and correlation ROC measures, and 20 new Bayesian tables were added for assurance primarily. And then the autumn release, for those who aren't aware, will include 32 new tables in the pre-existing set, 20 new tables related to proportions data and crossover designs, 12 new Bayes tables related to assurance, and then 15 new adapt tables in a new adaptive module or adapt module related to conditional power, additional group sequential designs options, and unblinded and blinded sample size re-estimation options. We're also including an undo redo option for anyone who's a long-term use of nQuery that will be available as well. So even if you ignore all the new tables, anyone who's been asking for undo redo, that's actually been included in that update as well. You'll find that out. You'll find all of that and other UI UX improvements in the release notes as when they come out. So just once again, just want to apologize for overrunning a little bit. But if you have any questions you want to ask me at a later date, feel free to email statsols at info@statsols.com, and I'll probably reply to those in person. And if you have any questions now, just include them in the questions tab of the webinar software, and I'll be happy to answer those now. So I'm just going to take a few moments to see what questions came in during the webinar and just answer those in a couple of moments. So I'll talk to you in like basically 20 seconds or so. The references are available at the end of the slide set, by the way, if you're interested. So there's two questions here, one from Debbie Anderson. I think she's left, but for anyone who's interested, why is the total success rate considered a nuisance parameter and not considered the effect size? And the overall, if you assume the original effect size is true, so you take the one that you used in study planning originally, you can then reverse engineer, given the overall effect sizes, what the actual per group effect sizes would have been in that situation. So you're not finding the actual effect size, you're just assuming that the original effect size used is true and then creating updated assumptions about what the most likely, well, what the group 1 and group 2 proportions should have been if your effect size, like your difference in proportions or odds ratio, was actually true, but the overall rates in each group was slightly different from what you expected, and what effect that might have on the sample size. I would say it's probably like less useful or less common because you have to make that very strong assumption about your original difference in proportions or odds ratio being true than the means case, but it seems to have some value as well. So there's another question saying, is it the overall power design, the power entered table, is it the overall power design using the blinded sample size re-estimation, or is it the target power? Sometimes we want to compare the overall power of this adaptive design versus fixed design. So what I would say is that for the blinded sample size re-estimation, in that case, what I would say in that case is that in the blinded case, you expect your overall power for a blinded sample size re-estimation and internal pilot approach to be the same as your initial power. So that's the same power for both of them. If you could obviously do a simulation if you wanted to compare that and make sure that's true, but the general idea is that the power for the interim analysis, if you're doing a blinded interim analysis, that is, you're not looking at the actual effect size and using an internal pilot approach, for example, the original power you target and the updated power is the power that you actually, like, it's the power you expect. Now you could reverse engineer and go back, for example, you could, like in our example here, you could like we have our updated internal pilot sample size for estimation of 17. So we'll set this to 16. And for fixed sample size, because obviously it's 68. So what was the worst case scenario if we just done if we just gone back and used 68, but the actual variance was higher than we expected? You can see that there was a reduction in power here of about like 6.5%. So that'd be pretty easy to do kind of manually. If that's something you're interested in or something that comes up commonly, then I'll be happy to perhaps include that as an option or as a side table in the either this release or in a future release. But that's a great question, actually. Thank you. There's no other questions, just except that someone was asking the slides. The slides will be sent automatically to you in an email later today, but you can still actually, with the last few moments here, go to the handouts tab of the webinar software and download them now. I'll leave this open just for a couple of minutes here with this in the background to allow you to do that, Rashida. So anyone who wants to download the slides right now, they're available under the handouts tab to download, but you will get an email later on today which will have that and the recording of this webinar. And there's one note just to slow down at the beginning. I hope I probably speeded up a bit towards the end, but I hope for the majority of it, it's been understandable for both of you here. So as I said, I'll just leave the slides open here for a little bit longer for anyone who's interested to download, but I'll be shutting down the webinar in about a moment or two. So I just want to thank you all for attending. Apologize for running over a little bit, and I hope you have a very good day and that you're interested in the adaptive features to be available in nQuery. And obviously this is just the start of what we're doing in terms of adaptive design at nQuery. We'll obviously be looking at other things. So if there's any type of adaptive design or sample size for adaptive design that you're particularly interested, do let us know, because your feedback is a very highly weighted component in the decisions about what we end up prioritizing for development time and research time. So thank you once again. I'll be ending the webinar in a minute. If you want to download the slides, they're available under the handouts tab.